So thank you for the introduction, Kate. It's my pleasure to be here today to speak about our TyphiNET Online AMR surveillance dashboard. So firstly, I just wanted to acknowledge the really large number of contributors that have made this project possible. In particular, I'd like to thank members of the Global Typhoid Genomics Consortium who have put a tremendous amount of effort into helping us develop visualizations that are representative of trends in their settings, for curating data and being willing to collaborate with us on this project. I'd like to thank the entire team that's been involved in this project, particularly Louise and Vandana, who have been our software developers on the project. So TyphiNET concerns surveillance of typhoid fever, which is a global health problem. It's an infection driven by the bacterium Salmonella Typhi, which is gram-negative and a serovar of Salmonella enterica subspecies enterica. And importantly, it's a human-restricted pathogen. We aren't aware of any environmental or animal reservoirs for this particular bacterium. And it causes an infection that's known as typhoid or enteric fever. And this is a systemic infection that drives more than 10 million illnesses annually, of which more than 100,000 are associated with mortalities each year. The infection is transmitted fecal-orally, usually through contaminated food or water. And as a consequence of this, it's endemic to many low to middle income country settings where there are insufficiencies in hygiene and sanitation related infrastructure for controlling outbreaks of waterborne diseases. The control of typhoid is complicated by two major factors. The first being asymptomatic carriage, and if you've heard of Typhoid Mary, you're already familiar with this. So Typhoid Mary was colonized by typhoid, shedding Salmonella Typhi in her stool and contributing to onward transmission of disease, but it was actually a healthy individual. And because of these reasons, typhoid carriers can be quite notorious to identify. The second complicating factor is why we're meeting today, which is antimicrobial resistance. So to just give a very, very brief overview of typhoid and AMR, it's a similar picture to what we know to be true of many pathogens. So for every sort of drug that we have shown has been efficacious for controlling Salmonella Typhi, resistance has emerged a short time afterwards. And in the years preceding the 1970s, efficacy for three drugs, often referred to as classical first-line drugs for controlling typhoid, was demonstrated. And this was chloramphenicol, ampicillin, cotrimoxazole. And following widespread multi-drug resistance to these three first-line classical drugs in the 1980s onwards, this prompted a change in treatment practice in many parts of the world to the use of fluoroquinolones in the 80s. And about 10 years later, we were seeing outbreaks of fluoroquinolone non-susceptible typhoid. And in the subsequent years following this, there were several sort of changes in treatment guidelines recommending azithromycin, third-generation cephalosporins, and in some cases carbapenems as well. We've subsequently seen resistance to all of these emerge in the pathogen population. And of particular concern is what's known as extensively drug-resistant typhoid. And this is sort of an ongoing situation in Pakistan, where typhoid strains there have become resistant to all oral antimicrobials except for azithromycin. And this is actually a major issue for typhoid because it places a lot of, should resistance to azithromycin resistance emerge, it will require healthcare facilities in low resource settings to start treating typhoid patients as inpatients rather than outpatients. So while antimicrobial chemotherapy continues to be threatened by the emergence of antimicrobial resistance, we do have other ways of controlling typhoid. There is a typhoid conjugate vaccine that was pre-qualified for use by the World Health Organization in 2018. And following successful efficacy trials, is being rolled out in a number of endemic settings globally with support from Gavi. And it's an improvement over previous generations of typhoid vaccines because it can be used in infants 6 months of age and above and is highly efficacious. However, improvements to water sanitation and hygiene infrastructure would serve to also control typhoid as well as many other waterborne illnesses. So the TyphiNET dashboard concerns the use of genome data for the surveillance of drug-resistant typhoid. And I'm sure many people are familiar with why this is, the desirable choice in this instance. A major driving factor for us is that for many settings worldwide, there's actually more genome data available for typhoid than publicly available AMR phenotyping data. And as sort of, sequencing costs decrease and bioinformatics expertise becomes increasingly more available, Genomics is increasingly being used as a core method of pathogen surveillance, both in research and public health laboratory contexts. We also know that AMR characterization via genomics is robust, it can be reproduced, and it's informative not only on AMR, but many aspects of pathogen biology, including virulence, transmission dynamics, and pathogen spread. And we know that genome data are readily stored in INSDC databases. They can be retrieved quite readily from these. And as our knowledge of AMR and genotype sort of frequencies and links become better understood, we can always go back and reinterpret the data as this knowledge and the biology of AMR, the genetic determinants driving this continues to develop. So what we set out to do with TyphiNET was to develop a dashboard that united the available whole genome sequencing data in the public domain for typhoid and placed it into an accessible and open online interface or dashboard that didn't require any prior knowledge of bioinformatics or pathogen genomics. We wanted to make this dashboard create intuitive, user-friendly visualizations of genotype or lineage frequencies and antimicrobial resistance prevalence. And we wanted this to be queryable both by location and time to investigate not only global trends like you can see in the visualization I'm showing here, but also national annual trends in antimicrobial resistance as well. And we wanted all of the data and visualizations to be downloadable by the user to either follow up on sort of specific genomics questions or genomic epidemiology questions, but also so that the plots and reports generated from this platform could be readily reused for communication purposes like presentations. And it's our hope that creating a user-friendly interface to genomic data that removes some of the requirements of bioinformatics knowledge and genomics knowledge will help make these data more visible to policy decision makers in terms of how they might guide empirical therapy, sort of target, control, and intervention strategies, and ultimately make public health policy decisions. So getting from having the sequence data and the metadata available to building a dashboard that can be used to inform policy decision making isn't a straightforward process. There's actually a number of different sort of infrastructural and software challenges that come along, as well as microbiology and data contributor challenges. So the picture and pathway that we've used in creating TyphiNET really looks something like this. Data contributors are routinely depositing whole genome sequence data and metadata for typhoid into INSDC databases. You then need a way of stratifying the population into meaningful subgroups, such as pathogen lineages, and you need tools that allow this to happen, such as multi-locus sequence typing, serotyping tools, and genotyping tools. You also need a means of calling antimicrobial resistance determinants from the genome data and interpretation criteria for predicting phenotypes from these data. You also need platforms that can house the processed data and process it as well. And at that point, you can get to the point where you can design an interactive dashboard to serve this up to a general user base. But at each of these points in the process, a number of challenges arise. As many of us know, metadata can be incomplete and public data can be heterogeneous, and it's difficult to understand which data might be representative of trends in the pathogen population. For the various sort of tools available to understand and stratify and sort of understand pathogen populations and to interpret AMR data from genomes of these is varying for different pathogens as well. And in terms of sort of databases that are available that can serve this information up for other purposes, the content and functionality of these does tend to differ somewhat. So I just wanted to walk through some of the challenges we had to address in establishing this dashboard. So the first one was missing metadata. And what we had to do to address this was to go to the community. So several years before we produced this dashboard, we brought together the typhoid genomics and medical microbiology community in the form of a consortium model. So this is the Global Typhoid Genomics Consortium, and I'm pleased to say that we are more than 200 members strong today, and we have representatives from over 50 countries. And we brought the community together to encourage data sharing of typhoid genome data for public health benefit as early as possible in an individual's work frame and an organisational's work frame. We wanted to provide tools and infrastructure to facilitate the extraction and reporting of key data of public health relevance on AMR and pathogen genotypes or lineage frequencies. And we wanted to promote and facilitate the dissemination of genome-derived data for monitoring antimicrobial resistance and, of course, post-vaccination impact. So part of the work we did as the consortium was we established a metadata template and standards for typhoid genome data. There are a number of fields in here that, as you'll see, are quite useful for the dashboard itself, but also serve to facilitate data reuse in genomic epidemiology studies. And the field I'll draw your attention to right now is this purpose of sampling field. And this is really focused around understanding what the biases are for typhoid genome data contributed by members of the consortium to this effort. So the point of this is really to understand which data we can consider non-targeted, and that is data that is likely to be representative of trends in the pathogen population more broadly. So these are data that are randomly subsampled from a larger ongoing surveillance study. They're non-targeted data routinely collected by reference labs, usually from trouble-associated cases of typhoid. All these emerge from routine diagnostics. Whereas targeted sampling is data that is enriched for, say, AMR or a particular cluster involved in an outbreak. And by having this field in there, we can understand how data can be used better in future studies. The second sort of challenge to establishing a surveillance dashboard that I mentioned earlier is sort of the typing tools available for a given pathogen. So in the case of typhoid, we're able to leverage previous work that we had done in establishing a genotyping framework. And what this is essentially is a nomenclature that is phylogenetically informative and provides a 3-digit or more address that allows you to rapidly type isolates according to pathogen lineage. And at a glance, when analysing a large data set, this tool actually allows you to identify, say, genotype 431, which is known to be associated with multiple intercontinental transmission events, and drug resistance at a glance when you're analysing a new data set. And the software associated with Typhi, this genotyping framework, has been through several iterations, but in the most recent iteration of this, we've worked with Sam Iqbal's team at EBI to integrate this into the microbe platform to allow typhoid typing direct from reads. And we've also included in this antimicrobial resistance typing and detection of mobile elements associated with spreading antimicrobial resistance. And we're fortunate in the case of typhoid that when it comes to understanding how AMR genotypes relate to AMR phenotypes, that this is a pathogen where we have a really good understanding of this. And I'm just showing a table from a paper from Marie Chataway and colleagues published in 2021. This is data from the United Kingdom Health Security Agency that demonstrates when we compare AMR genotypes to AST testing phenotypes that we see 99.9% concordance. So we have a good knowledge of how to interpret genomic data for typhoid in terms of AMR. And the last challenge I highlighted was database content and functionality. So you have the data, you have the tools, but how do you actually process this in a way that can then be drawn into a web interface regularly and updated regularly as well? So in the case of typhoid, we've been working with colleagues at the Centre for Pathogen Genomic Surveillance now for quite a long time to integrate our genotyping framework into Pathogen Watch. And if you haven't come across Pathogen Watch before, I highly recommend it. It's a really, really useful web interface that allows users to drag and drop genome data into any standard web browser and carry out a genomic epidemiology study. So this will allow the individual to infer a phylogenetic tree, to type the isolates according to multi-locus sequence typing and our genotyping framework, and to also detect molecular determinants of AMR, so be those genes or mutations. There's also a vast amount of contextual data that can be used to make global context trees to understand global and regional transmission dynamics as well. So this became sort of our hub in this instance for typhoid data. So all data that is curated by members of the Global Typhoid Genomics Consortium using that standardized metadata template we created, then gets entered as collections along with that metadata and the genome data and the genome-derived data in terms of genotypes and AMR into this platform for future use by the community. So this gets us to the point where we can start addressing this really as a software problem. So I just wanted to reiterate the sort of data flow that TyphiNET uses. So individual data contributors deposit their genome data into INSDC databases like NCBI and ENA. Global Typhoid Genomics Consortium then downloads those data, assembles those in a standardized manner, completes and curates the metadata associated with those, including sort of the primary fields we're interested in for this dashboard, which is country, whether it's a travel-associated case, and if that is the case, where that travel-associated infection was acquired, the year of sampling, and whether this can be considered a sample that is non-targeted or representative of pathogen trends. These are then uploaded to Pathogen Watch, which carries out genotyping and antimicrobial resistance calling. And those genome-derived data and metadata are then pulled into TyphiNet via an API called Spider that just draws down the publicly available files from that system. This is then injected into our dashboard, which is, in terms of its software architecture, a MERN stack JavaScript application with MongoDB powering our own database. And I should mention that the code for this is freely available by GitHub. So when you first navigate to typhi.net, what you tend to see is this global overview of typhoid and AMR. So by default, this is showing you ciprofloxacin non-susceptibility as a global map that is a heat map, where the sort of more intense and darker shades of red indicate a higher prevalence of resistance to ciprofloxacin. And you're not limited to just ciprofloxacin. There's a drop-down menu here that allows you to select a number of different antimicrobials that are relevant to the treatment of typhoid, but also to explore the data in terms of genotype prevalence if there's a particular lineage that you're interested in understanding the global spread of, for instance. If you hover over different countries in the map, you do see tooltips which provide some summary statistics. In this case, we're looking at ciprofloxacin, so it provides the percentage of cases from Pakistan that are non-susceptible to ciprofloxacin. But these tooltips change depending on which drug class and sort of data view you're exploring. You also have the option of filtering data by time and by source. So, for instance, you might want to use the drop-down menus here to look at data from the most recent 5 years available, or you might like to filter to just locally collected genomes from a given setting, or travel-associated cases sequenced in high-income countries where cases of typhoid are notifiable. In that case, those will be viewed by the country of origin. So if an individual's typhoid case was sequenced as a part of routine public health surveillance in the United Kingdom, but they had picked up their infection in India, the sample would appear as a sequence from India. And with all of these visualizations created by the dashboard, there are little camera icons next to all of the plots that allow the user to download the plot they've generated as a PNG file for onward use in presentations and reports. So if we think of Pakistan as a case study, looking at this global overview, what you can do is click on Pakistan in the map, which then triggers these sort of country-specific plots down the bottom here. And these can be filtered by time using the filters that I just showed previously, and also by local or travel-associated cases. So when you want to explore country-level trends, there's a number of different plots that you can view. One of these that I think is quite helpful is this drug resistance trends plot. And if you take a look at this particular example, what we can see here is that these purple lines actually increased from 2016 onwards. And what they're showing is increasing prevalence of resistance to third-generation cephalosporins, as well as ciprofloxacin, which are key drugs involved in control of typhoid. What's really interesting when you compare this to the lineage or genotype distribution plot is you notice that this black genotype, this 4311P1, according to our genotyping framework, increases in prevalence over time in this same time frame. So it appears that this increase in these resistances is associated with this particular pathogen lineage or genotype expanding clonally in Pakistan. To link this up even more tightly, what you can do is then you can then look at resistance frequencies within genotypes. And this is a bar chart showing the sort of percentage of samples within a given genotype or lineage that are resistant to a given antimicrobial. And what you can see here is that these purple bars that indicate resistance to third-generation cephalosporins and ciprofloxacin are mostly associated, almost entirely associated with this 4311P1 genotype. You can then look at this resistance determinants within genotypes plot, and you have the option there of selecting a particular drug class that you're interested in. In this case, this is looking at third-generation cephalosporins, and we can see that this resistance to third-generation cephalosporins has been driven by the acquisition of a BLA-CTXM15 gene by this particular genotype shown in the blue bar plot on the right here. And what this is representing is the emergence and subsequent expansion of extensively drug-resistant typhoid in Pakistan that is resistant to all oral drugs except for azithromycin that I was alluding to earlier in the presentation. Another helpful feature of TyphiNet that I just wanted to highlight here is that we have made it so that you can download not only the individual plots with these little camera icons that are next to every sort of plot that you can see in the dashboard, but we've also made it so that you can download a line list of the underlying data using one of these buttons at the bottom here on the left-hand side. This is a plain text CSV format that is a line list of all data available. It includes not only the genome-derived data on AMR, but also genotypes, the metadata that explains the sampling, data contributors by name, any publications associated with the data set in terms of DOI or PubMed ID, and also contains the accession numbers for the sequences that allows users to explore further trends they might have noted in the data, either using Pathogen Watch or their own bioinformatics setup. It also allows sort of tracking of data provenance as well and individual data contributors. The other download button you can see at the bottom there triggers the download of a PDF report, and this captures all plots a user has generated in a given setting. The sort of splash page or title page of this report provides information on when it was generated, where the data have come from. It also provides information on the publications that have that the data are drawn from. It also provides some variable definitions and links out to, say, the genotyping framework, various explanations of how we handle travel-associated cases in the dashboard, and provides sort of information on how we've defined antimicrobial resistance in the dashboard itself in terms of the rules that equate to the sort of phenotype predictions that we make in this platform. So in conclusion, what I hope I've been able to show you today is an open and centralized resource for typhoid genomic surveillance that doesn't require expert knowledge in bioinformatics or genomics. It provides global overviews and country-level summaries of AMR trends over time and geography to help guide empirical decision-making, be that in country itself based on resistance frequencies or based on location of travel where an individual has acquired their infection overseas. We hope that it has utility in targeting and monitoring control and intervention strategies and helps make these data more visible to policy decision makers. I just wanted to highlight that this isn't the end of the road. We've established this TyphiNet platform as a pilot sort of pathogen for Salmonella Typhi, but you're also working on expanding this platform in AMRNet to encompass a number of World Health Organization priority pathogens. So again, I'd like to thank all members of Global Typhoid Genomics Consortium who contributed to this work, and also like to thank the entire team, in particular Louise and Vandana, who have been our software developers on the project. And thank you again for the opportunity to share this with you all today. I'd be happy to take any questions at the end of the session.